Hello people, this is a short presentation on carbohydrate anabolism. We must know what is metabolism. Now metabolism can be simply defined as the series of chemical reactions which are essential to sustain life. So what will we talk about? We will be talking about gluconeogenesis, glycogen synthesis and Coli cycle. And all this we will be talking with respect to anabolism. Now, gluconeogenesis. Now, gluconeogenesis is an anabolic pathway that results in generation of glucose from non-carbohydrate substrates. In simpler terms, we can say that gluconeogenesis is exact opposite of glycolysis. When we talk about gluconeogenesis, we are simply talking about how can we convert pyruvate into glucose. Now this is a very simplified diagram of gluconeogenesis pathway in which glycerols, amino acid and some components get in this way to overcome some irreversible steps of glycolysis in order to reverse the glycolysis reaction and obtain glucose. As we see to the name, gluconeogenesis, which means genesis means production, neo means new, and gluco means glucose, which means making new glucose. Liver cells can do gluconeogenesis, but in order to obtain glucose from pyruvate, we need to overcome some irreversible steps in the glycolysis. Before we get with the reactions, we must know the background. First of all, gluconeogenesis is universal, that is, it is available, or that is, it is functional in plants, animals, fungi, bacteria and other microorganisms. It takes place mainly in liver, to lesser extent cortex of kidney, intestine, muscles and some multisynaptic neurons or astrocytes of the brain. The process is highly endogenic, that means it takes energy to come up until it is coupled up with the hydrolysis of ATP or GTP, effectively making the process exogonic, which means it gives energy. Getting over, essentially, the gluconeogenesis is only possible if we can get over the three irreversible steps of glycolysis. Here is a diagram depicting the location of gluconeogenesis, the entire Cori cycle, and an overall chemical equation for gluconeogenesis. Now, if we remember glycolysis, the glycolysis consists of various steps, but among them, the first irreversible step was the formation of glucose 6-phosphate from the glucose. Then it was isomerized to fructose 6-phosphate. Then the next irreversible step was the conversion into fructose 1,6-bisphosphate. Then there were many steps, and then after those steps it finally converted into PEP, which in turn gives the pyruvate. So let's have a close look. Here we are having two cells, either it is muscle cell or some other cells of the body which are involved in glycolysis, or we have here the liver cell or the hepatic cell which are responsible for gluconeogenesis. Between them we have bloodstream. So the bigger the piece of paper you have, it will be easier to understand. Now when we give a closer look to the diagram, we can see that here we have muscle cell, here we have muscles and other cell, the liver and hepatic cell, the cytosol of liver, mitochondria and the matrix. Let's bring in a quick recap. If we have a closer look to this diagram, we have a glycolysis reaction over here. I am leaving up all the various steps which were involved in glycolysis and focusing on the main steps that we need in gluconeogenesis. For the reference in gluconeogenesis, here we have glucose which is converted into pyruvate, and further with the fermentation it got converted into lactate or lactic acids. So when this lactic acid enters the liver cell, it's converted into pyruvate. We have pyruvate. The pyruvate gets into the mitochondria matrix, which is converted to oxaloacetate. If you give a brief look to this reaction, you will remember that this is the part of TCA cycle. Once we have pyruvate, we will try to obtain glucose from the pyruvate. Once we have pyruvate into mitochondria, we convert pyruvate into oxaloacetate. Now if you remember, pyruvate is a 3-carbon molecule and oxaloacetate is a 4-carbon molecule. So here we are adding a bicarbonate. We are adding up a bicarbonate which is equivalent to CO2. Doing this, we need energy. In order to convert pyruvate to oxaloacetate, we need energy, and this energy is obtained by utilizing our ATP. So here we will be using the ATP hydrolysis to enhance the chain from 3-carbon to 4-carbon. This is ATP hydrolysis. The enzyme that we use in this step is called pyruvate carboxylase. Once this step is done, it is shipped out from mitochondria to the cytosol of liver. Now here oxaloacetate is converted into PEP. Now as we know that OOA, the oxaloacetate, is an 4-carbon molecule and PEP is a 3-carbon molecule. We lose 1 carbon in form of CO2. And the enzyme which is responsible for doing this step is PEP carboxy kinase. Once we are done with this step. Once we are done with this step, the reaction is further carried out to fructose 6-phosphate. Now, conversion of phosphophenol pyruvate to fructose 6-phosphate is catalyzed fructose 1,6-phosphatase enzyme. Till here, we have utilized two ATP equivalent ATP, and another one was GTP. Once we are done with the formation of fructose 6-phosphate, it is reversibly converted to glucose 6-phosphate. Now, similar to the previous reactions, we know this phosphate is removed by addition of a water molecule, and glucose 6-phosphate is converted to glucose. Once this glucose is converted, it is given out or shipped out from the endoplasmic reticulum, and this glucose is shipped out from the liver cell. And this reaction is catalyzed by glucose 6-phosphatase enzyme. Now this glucose can get into the bloodstream. And if any other cell needs glucose, it can enter into their cell. Now, to give a broad view of this entire cycle from glucose to pyruvate to lactic acid, and then from pyruvate to glucose again, this whole cycle is called Cori cycle. Now, the pyruvate is converted to oxaloacetate using pyruvate carboxylase. Then this oxaloacetate is converted to phosphophenol pyruvate using PEP carboxylase kinase. Now there is a whole bunch of reversible reaction, as you can see here, which in end gives us to fructose 6-phosphate. Further, fructose 1,6-bisphosphate is converted to fructose 6-phosphate using fructose 1,6-bisphosphatase. Now fructose 6-phosphate is converted to glucose in a reversible reaction. By paying a closer look, we can see that the glucose 6-phosphate is converted to glucose using glucose 6-phosphatase. Now, this is the overview of how this reaction takes place. This was the whole reaction, and you can note down the steps as given in the slide. Another process in carbohydrate anabolism is glycogenesis or glycogen synthesis. Now, glycogenesis is a process of glycogen synthesis in which glucose molecules are added up to build a chain of glycogen for storage. The process is activated during the rest period in Cori cycle. Before proceeding to the glycogen synthesis, we must know what glycogen is. Now, glycogen is a polymer of glucose. If you break down the word glycogen, you will find that gluco and gen. Gluco means glucose and gen means production, which in short means glucose producing polymer. It is primarily used for storage of glucose. It can be utilized when glucose is needed. Glycogen is a large polymer of glucose monomer, and this monomer can be either arranged in linear or branching fashion. The linear linkages are alpha 1,4 and the branching linkages are alpha 1,6. It is a rapid source of glucose for the body. Now if you pay a closer look to the structure, you will find that there is a protein structure in the middle of the glycogen molecule. This is glycogenin. This facilitates autoglycosylation by adding up 10 to 20 glucose molecules. Glycogenin is an initiating enzyme. It helps the autoglycosylation by initially adding up 10 to 20 glucose molecules to the glycogen chain. Now, if we pay a closer attention to how glycogen synthesis happens, it all starts with glucose. When glucose enters the cell or is produced within the cell, it is converted to glucose 6-phosphate using glucokinase or hexokinase. It is simply phosphorylation of glucose at the 6th carbon position. Now the glucose 6-phosphate is converted to glucose 1-phosphate, which is simply shifting of the position of phosphor group from 6 to 1. This is made possible by phosphoglucomutase enzyme. Once we have glucose 1-phosphate, it is acted upon by UDP glucose pyrophosphorylase, which is activation of glucose. It is very important step as it adds up the UDP to glucose, which results in the activation of glucose. Once this UDP glucose is obtained, it is added into the chain of glycogen. This is done by glycogen synthase. It simply removes the UDP and converts into a non-reducing glycogen chain. Branching and linkages are catalyzed by glycogen branching enzyme. One of the most important enzymes here is glycogenin. The glycogenin is responsible for autoglycosylation. It adds up nearly 10 to 20 glucose molecules in order to start glycogen synthesis reaction. Talking about glycogen synthesis mainly happen either in the liver cell or the muscle cell. Now talking about the liver cell, its location is cytoplasm, and even in muscle cell the location is cytoplasm. The main role in liver cell is to maintain blood glucose level, so whenever the glucose is needed in the blood, the glycogen is broken down to simple glucose and released in the blood, while in the muscle cell its role is to provide the quick energy source to the muscles. The glycogen stored in the liver cell is used during fasting, and in muscle cells it is used during exercise. The stock of glycogen generally lasts for 24 hours in the liver, while the glycogen reserves in the muscle cell is rapidly consumed and also synthesized.